In Chapter 8, we are taking a look at microbial metabolism. And Chapter 8 starts off with some information about what energy is and enzymes which are necessary for metabolism to take place. So, when we think about the word metabolism, a lot of times we think about it in terms of, oh, my metabolism's slowing down as I get older, I don't burn calories as fast, or, oh, someone has a high metabolism, they can eat all they want to and never seem to gain any weight. So we use metabolism in that way, but really, metabolism is just the term that we use to describe all of the chemical reactions inside of a cell. So all of those different chemical reactions that are taking place within all of those different organelles that we've talked about, that is considered metabolism. And there are two major types of reactions that are going to take place in metabolism. And we have anabolism, which are endergonic, meaning they require energy to proceed. And anabolic reactions are going to convert smaller building blocks into more complex molecules. So anabolic reactions are building reactions. They're going to take something small and use those to build something large, like taking amino acids to build proteins. On the other hand, catabolism refers to exergonic reactions. These reactions release energy, and these are a breakdown type of reaction. We're going to take something large, like that protein, and break it down into its individual components, like those amino acids. It takes both to come together to equal our metabolism. And so in this graphic here, you can see where we have, anabolism, we have to use energy. So energy has to be input, and that's usually going to be in the form of ATP, which we'll talk about a little bit later on. And that drives our anabolic reactions. And then we have those catabolic reactions where we're breaking down those molecules into smaller building blocks. And a lot of times these are going to be recycled and then used to build again. Now, when we look at our classification of organisms by energy and carbon source, we're going to see four major classifications here. We see our chemotrophs, and we divide those into chemoautotrophs and chemoheterotrophs, and then phototrophs into photoautotrophs and photoheterotrophs. So chemo referring to chemical, where they're going to get their energy source. Photo means light, so the energy source is coming from sunlight. Auto and hetero here, auto means it's self-made, and then hetero means that it relies on others. So if we look at the carbon source here, we'll see inorganic versus organic. So inorganic are those that don't contain carbon. And then we look at organic compounds. These are large compounds that contain carbon, things like carbohydrates or proteins, nucleic acids, fats, steroids, things like that. So a chemoautotroph relies on chemicals for their energy source, but their carbon source is going to be inorganic. And so if we look here, what types of examples are provided, these are sulfur bacteria, bacteria that can use iron and nitrogen. Then if we look at our chemoheterotrophs, we see all animals, and then most fungi, protozoa, and bacteria. So most of the organisms that we're going to talk about are going to be chemoheterotrophs. Then if we look at our phototrophs here, all plants are photoautotrophs because they can make their own food using carbon dioxide from the air. Carbon dioxide is an inorganic compound. Algae the cyanobacteria, and some of our green and purple sulfur bacteria are all photoautotrophs. And then our green and purple non-sulfur bacteria and our heliobacteria are photoheterotrophs. Their energy still comes from light, but they rely on organic compounds like we do for their carbon source. So make sure that you refresh over this. Now, when we look at these metabolic reactions, we have coupled reactions called redox reactions. Redox reactions are coupled reactions where we have two simultaneously occurring reactions. And redox is a shortened form of oxidation and reduction. So when we look at metabolism, and especially as we get into cellular respiration, one of the main drivers for these reactions is the transfer of electrons. Most of the energy that is stored within atoms and therefore within these compounds, and the energy that we need to fuel those anabolic reactions, are in the form of electrons. So we're going to strip electrons away from some compounds and add those to other compounds. And that's what redox is. Oxidation is when we remove electrons, and reduction is when we add electrons. I know it sounds counterintuitive if you think reduction like, oh, we're losing electrons, but that's not the case. So just kind of clear that out of your mind. Oxidation reactions, these molecules become oxidized because we're removing electrons from them. And when they're reduced, we say that they are accepting, or we're adding electrons to them. And these reactions must occur together. When we strip away electrons from one molecule, we have to have another molecule that we can add those electrons to. So that's why they're coupled. They occur together, and we shorten that to redox reactions. Now, when we have those catabolic reactions and we're breaking down those nutrients, we release energy, okay? They're exergonic reactions. And we can store that energy in the form of electron carriers, the reduction of our electron carriers, so adding electrons to specific electron carrier compounds, or in the bonds of ATP. ATP is the cell's usable form of energy. Okay, it's like the cell's currency. Okay, and in living organisms, we have a group of molecules that function as these electron carriers, and they are mainly B vitamins, and they are NAD, NADH, and FAD, and this is what they stand for there, but we don't, we're not going to use their whole names, just NAD, NADH, FAD. And these compounds are very easily oxidized and reduced. So it makes them perfect to be electron carriers. We can add electrons to them, and they can carry these electrons, and then when we need them, we can easily take them from them. I kind of think about these energy carriers, NAD, NADP, NADH, FAD, as Uber drivers or cab drivers, in that they pick up passengers, which are electrons, they carry them where they need to go, and then they drop them off. So they're kind of like Uber drivers in that way, that they will show up in areas where these catabolic reactions are occurring, where these electrons are being stripped away. They'll latch on to those electrons. They bond very easily with those electrons, and then they carry them somewhere else in the cell where they're needed, and they drop them off. So they're very easily reduced, picking up the electrons, and oxidized, dropping off the electrons. NAD and FAD are used they're used very extensively in catabolism in chemoheterotrophs. NADP, NADPH, we more often see in photosynthesis for those, phototrophs that we talked about a while ago. Now, ATP is the cell's form of energy. Okay, so ATP primarily comes from glucose. We can make ATP from other organic molecules and like fats or proteins, but we have the most efficiency and we get the most ATP when we break down glucose. And that is the process of cellular respiration, which we're going to get into. But during this form of breakdown, in this form of metabolism here, we're going to produce ATP. Now, ATP is the cell's form of energy, and you can kind of think of ATP like a rechargeable battery, okay? That when we get solar chemical energy that's going to drive these reactions, we undergo a process called phosphorylation. And phosphorylation is a fancy term that we use to say that we're adding a phosphate group, okay? This is like taking your rechargeable batteries and putting them in the charger, okay? We're recharging this molecule, essentially. And then ATP can now be used. Just like that battery, it's recharged. Now it can be used to power a flashlight or whatever you need it for. But then as it does work, it loses those phosphate groups. It essentially loses its charge. Okay, and that's the process of dephosphorylation, where we're removing a phosphate group. The energy in ATP is stored between these bonds, between these phosphate bonds. So as we strip away a phosphate, we're releasing a lot of energy. Okay, so as that battery is doing its work, it's essentially releasing energy in some form. And that's exactly what ATP does. So the cell has to have ATP in order to perform all of its different reactions. Okay. And the recharging process doesn't use as much energy. And that's why it's an effective process. All of these chemical reactions in these metabolic processes require a catalyst, something that's going to speed up the reaction. And in living organisms, we call those catalysts enzymes. There's a special group of proteins that function as catalysts. They basically control cellular metabolism. Without enzymes, these chemical reactions that we need would proceed way too slowly for us to survive, whether we're talking about humans or we're talking about bacteria. So we have to have these enzymes in order to speed up reactions. And what enzymes do is they lower the activation energy. Activation energy is the energy that's needed to begin a chemical reaction. And so enzymes lower that amount of energy. And in this image here, this figure, you can see we've got our reaction path, so how our reaction is going to proceed on our x-axis, and we've got energy going up here on our y-axis. So without a catalyst, you see our activation energy is way up here. We need a lot of activation energy. But with a catalyst, the red dotted line, we've lowered that amount. It's kind of like if you're pushing a rock up a hill, an enzyme would lower the height of the hill. Okay, so you're not going to have to use as much energy to push that rock up the hill. It's not going to take as long for that reaction to proceed. So that's what enzymes do. They lower that activation energy. And enzymes are very specific. They work only on a specific substrate or substrates, kind of like a lock and key. Your front door has a key that fits into it, and that key doesn't work on any other doors, and other keys won't work on your door. So enzymes have specificity to certain substrate molecules.